My name is Paul Simonson. I'm a hematopathologist at Weill Cornell Medicine, and today I'll be speaking on applications of machine learning in clinical flow cytometry. So by way of outline, I'll talk a little bit about what is machine learning, how can machine learning help us in flow cytometry and specifically clinical flow cytometry. I'll talk a little bit about unsupervised cell clustering, basics of supervised machine learning, and I'll give a couple of examples of applications. So what is machine learning? So at the heart of machine learning is statistics. It's very good at identifying patterns. It learns what we teach it. So if we don't present examples to it of things we want it to learn, it won't learn it. So something to keep in mind. And at the heart, what we're trying to do is create models. And so one of the simplest models we can think of to describe data is just a straight line where we're fitting different parameters to the data. But in machine learning, we have similarly other models, but they're much more complex. There are two general categories of machine learning. So first we have unsupervised machine learning. So this is something where we don't apply labels to the data. We basically let the machine try to separate different populations from each other and so forth. And this has been used for things like clustering algorithms and dimensionality reduction for viewing data like UMAP. There's also supervised machine learning. And so some of you may have heard of some of these terms like random forests, support vector machines, and deep learning. These are all different, what we call classifiers or different algorithms used for supervised machine learning. Okay, so what machine learning is not, it's not a replacement for pathologists. It's not sentient robots. It's not the end of humanity. Okay. So how can machine learning help us with flow cytometry? So first of all, we're hoping for better accuracy. So can we reduce the subjectivity that we're using when we're interpreting flow cytometry data? Could it maybe help the pathologist by alerting us for possible misdiagnosis? Maybe we're going very fast in the afternoon, we're getting sleepy or something. Maybe the machine can come in and say, hey, maybe you missed that one. Can we use it to help us highlight the cells of interest while we're going through the data? Hopefully it can help us with a faster workflow. So maybe it can help us automate the ordering of additional antibody panels. So maybe the first screening panel comes in and it says, oh, there might be a B cell neoplasm. Maybe you should order additional B cell panels. It can help us alert us for high priority diagnoses. So maybe we'll look at those first instead of waiting till later in the afternoon if there's new acute leukemia present. And it could help simplify increasingly complex analyses and maybe pre-populate reports for us. And hopefully this all also results in a reduced workload. For some reason, people don't really seem to think that it will result in a shorter work week. Okay, so what are the specific opportunities we have? So in the general workflow in the clinical lab, first the specimen comes in and it has to be processed. We have to place orders, which are often based on the prior history of the patient, and we have to pick initial antibody panels. Then we flow the specimen, and then the data are reviewed by technologists who may order additional antibody panels and draft a report. And then it's reviewed by hematopathologists who may also do the same. So machine learning might help us with reviewing the prior history, maybe making the initial antibody panel choices for us, maybe picking those panels could help us draft the reports before we get to it. And same thing on the pathologist side. And in some cases, if it's a new diagnosis, we want another pathologist to look at it to confirm. Well, maybe at some point we reach in the future, the AI will be good enough that maybe that's our second pathologist who can help us sign the report. And hopefully, again, improvements in accuracy and ultimately improvements in clinical outcomes. So you guys from our talk so far already know very much about high dimensional flow data, but in the clinical lab, most labs are currently running 10-color flow cytometry. So initial application, let's suppose we have a new 18-color panel or flow cytometer, and we want to build a panel for this. And this is something we've been working on in our lab. So we're basically developing an immunomonitoring panel. And this could be helpful for looking at T-cell subsets in patients who maybe undergone bone marrow transplant and so forth. It might be useful for clinical trials and eventually use in actual clinical workflows. So we've decided we want to use computational methods in addition to traditional gating to help in the analysis. So why use new computational approaches? First of all, the increasing number of flow channels means there's increased complexity. So for us, we're looking at two-dimensional plots. That's a lot of two-dimensional plots that increases dramatically with increased channels. Increased numbers of gates can also lead to increasing chances of spillage of cells into the wrong gates. So for example, if we're looking for chronic lymphocytic leukemia, which is something characterized by CD19 positive, CD5 positive B cells, if we have a little bit of spillage into that gate, well, if someone's not very familiar with looking at the data, they might say, oh, look, we've got a small amount of CLL there. But if we're using newer computational methods, maybe we can help reduce some of those problems. Relying on traditional gating also results in bias that might help us or cause us to miss unexpected populations that might be there. And so newer clustering algorithms and so forth can help us avoid that. Okay. So some of the basic steps. First, we need to clean up the data. Then we do the clustering of the data. We visualize it and inspect the clusters and give them names. And then we might do further downstream analysis. So clustering algorithms are nice because instead of looking at a bunch of two-dimensional plots, they can look at all the data at once in ways that humans are not good at. And they can help us identify unexpected clusters. Okay, so there are very many clustering algorithms already out there. So this list here, you may think, oh, is that an 80s rock band list? No, this is just all the different clustering algorithms that have been published, and there are probably even more than this. Some of these clustering algorithms use different approaches. So some are supervised algorithms, which means that you're taking labeled data that you wanted to find the different populations that correspond to those labels. Some are completely unsupervised, so you're throwing the data to the algorithm and it's just separating it into populations. And then there's semi-supervised, so something in the middle. So how can we choose a clustering algorithm? So we want something that's accurate and reproducible, and similar cell populations are found in different specimens, and it's repeatable. Of course, it meets the needs of the problem at hand. And then another question is, are other people using it? So if somebody else is using it, it probably means that they've at least think it's good and useful. So in terms of comparing clustering algorithms, previously there was a FlowCap1 challenge, which was a detailed review of clustering algorithms. And it was very good, but it didn't contain high-dimensional data. Subsequently, there's this review of algorithms that has been very good in that it's compared high-dimensional data and has some variability in the types of data it's looking at both major cell populations and rare cell events. So what were the results of that? So in terms of all these clustering algorithms, one algorithm kind of stood out, which was FlowSOM, both in terms of accuracy and in terms of speed. And there have been other reviews since then that have kind of confirmed that FlowSum is a very, very powerful and fast algorithm. So FlowSum was first introduced in 2015, and it finds clusters in an unsupervised way. These cluster types can be applied to new cases. So basically what it does is it takes the data that you trained it on, and it creates what's called an embedding or map. And then for additional new cases, it can project the new data onto those embeddings or maps. So as opposed to things like TC and whatnot, where it might start over from scratch, FlowSum can look at new data. It's very fast. It can be run on most computers, even a lot of laptops can run it, and it's widely adopted. Some of the disadvantages, it might miss very small populations, and it does use a lot of computer memory. So how does it work? It works by creating a self-organizing map. For visualization purposes, it will also create what's called a minimal spanning tree. And then it also applies a consensus algorithm to the clusters that it's found to try and find the meta clusters, or the clusters that make up the overall populations, whereas the smaller clusters might just be small components of those. So the first step is generating the self-organizing map. And basically this works through an algorithm that creates a series of connected nodes, and it iteratively pushes these nodes around to find the data clusters until it's fit, and then that's the embedding that can be applied to additional cases. The number of nodes is usually chosen to be much larger than the number of cell populations we're expecting. And because of that, we then need to use what's called a hierarchical clustering or meta clustering to group some of those nodes together. Okay, so the higher the number of nodes, the more specificity for small populations we can see. These can be plotted using star plots, which basically gives us a visual way to look at what these individual nodes or clusters are composed of, whereas the different parts of the pie that are larger demonstrates more expression of a given marker. Okay. And again, for visualization, we can also plot what are called minimal spanning trees. Nodes that are most like each other are linked to one another, and they create basically these branches like trees. In a nice set of data from like bone marrow, you can actually see maybe the progression of maturation of cells through blast to myelocyte and metamyelocyte and so forth. And again, these nodes need to be clustered into what are called metaclusters to give kind of the bigger population cluster that describes those cells. Okay, so some of the practical considerations, of course, you want to minimize the pre-analytical variables, so minimizing batch-to-batch variability and using calibrations. You want to pre-process the data, so removing the non-viable cells, doublets, and so forth. Also applying the compensations and transforming the data. Some of those transforms will also involve choosing which markers are most important for the mapping you want to make. And you can also add additional weights to those markers. So say I want this marker to have twice the weight of another marker. You can do that before the step where you feed the data to the FlowSum algorithm. Okay, you also want to make sure that you have enough data and that it represents all the phenotypes you're expecting. So you may need to concatenate different files to represent that. And also you might want to create a pipeline so that you can just do this reproducibly rather than having to run it manually each time. And then there is at least one good tutorial online that you can find reference there. Okay, so how does one know the clusters are real? So one approach is you can try different clustering algorithms to see whether the same clusters are found via different approaches. You can also try the same algorithm again. So a lot of these run using random number generators. So if you started again with a different random number seed, it might come up with slightly different results. So if you run it multiple times, do you see the same clusters falling out? And of course, you want to visually inspect things. And you can apply dimensionality reduction algorithms to visualize the data. Go into this too much because I think most of the people in this audience are very familiar with t-SNE and UMAP and so forth. One thing just to note is that dimensionality reduction is not the same as clustering. So sometimes people get that a little confused. So t-SNE was developed in 2008. It maps high dimensional data to clusters in 2 dimensions. So it helps us. It does kind of cluster the data, but sometimes the clusters are split into different cell populations. Part of a population will be on one part of the t-SNE, whereas the other half is on the other side, just because they got pushed apart in early steps. And it's available in some commercial software. So UMAP is what I think many more people are using now, and which I also prefer. But it does a better job of preserving the overall relationships between cells. So kind of along principal component axes and so forth, you can think of kind of on larger scales and smaller scales. And it's also nice compared to t-SNE because the embeddings that it learns from one set of data can be applied to new cases. And there's a tutorial that's very helpful. Okay, some of again, the pros, you can apply it to new cases. And it's also good at representing the large scale relationships. I'll just skip this slide in the interest of time. Okay, so how can we implement it in a real application? So one thing we can do is set up a computer on a local network that's going out and actively looking for new FCS files as they're deposited on a server. So with those files, we can then run our UMAP or other clustering algorithms and create the labels, which we can then use as new channels in our FCS files, save those and review those with our standard flow cytometry software. So one way we've solved that problem is, again, we create the files, and in terms of creating the new channels, what we do is for every label or that we might make, we'll actually give it 2 coordinates, so an X coordinate and a Y coordinate. And what we're doing is we're plotting it on a grid. So we can actually just add a little jitter or noise to those coordinates. And so the dots are basically big enough for us to see in our flow cytometry software. And then we can just draw gates around those nodes in the grid. So we can basically just use all the functionality of the flow cytometry software that's already there. So it's just a simple way for us to switch back and forth between cluster label and our traditional gating. And so we can directly compare. Okay, so some additional considerations. You might consider lumping several cases into one and looking at how the data compare. Let's see, so one question is often, how many cells do I really need? So the answer is basically you need enough cells to adequately represent the immunophenotypes you're trying to find. What kind of computer power do I need? Again, a lot of this can be done on laptops and so forth. And should I hire a data scientist? So this answer, if you have a question in your mind about this, probably the answer is yes, unfortunately. But if you want to spend a few months learning, you can do that as well. There are a lot of additional software packages available. So getting to the clusters is just the first step for fancier analysis, you could say. So there are packages like Bioconductor, ScanPy, Seurat, and PathML, which I like to advertise because it's homegrown, collaboration between Dana-Farber and Weill Cornell Medicine. And then the last question is, who will sign the report? So is the hematopathologist ready to take responsibility for these clusters that have been found by the algorithm? And so the hematopathologist probably needs to become familiar with these methods and the underlying ways in which they work. Okay, so moving on to applications of supervised machine learning. So supervised machine learning is often referred to as a black box. And this is because there's so much complexity in the algorithm. And nobody likes a black box. So physicians don't like it, because we want to understand the data. And if we're going to make a diagnosis of cancer, we want to know what's going on. Machine learning practitioners don't really like it. And regulators don't like it. So FDA and so forth, they want to know, how is the machine making this decision if we're going to give it FDA approval? Okay, so how can we look inside the black box? So there are multiple methods for doing this. Some algorithms have been developed. They're kind of general use algorithms. So one is called CHAP, another is LIME, and there are other algorithms. And these kind of depend on which machine learning algorithm you're using in your choice of which one you want to use. But generally, CHAP and LIME, again, are kind of general use. This is the subject of many groups, and the methods are getting better all the time. So before we jump into that too much, I just want to talk a little bit about Flowbot. So I kind of call this our dumb AI, but it can actually do a lot of work. So machine learning takes a lot of development. It requires labeled data and so forth. Is there a poor man's AI that we can use in the meantime? And so just rules-based automated report generation is very helpful. We're using that a lot. Basically, and look at the analysis files that we've already done and create a template report that we can use in the lab. And it can also look at the data as it's coming in, create a list of cases we might want to prioritize. So maybe there's a new B-cell neoplasm or acute leukemia. It can create a text file that we look at as we go through the day and just prioritize our time. Some of the advantages compared to more complex machine learning is the algorithm is fully understood and completely customizable, but it's not very fancy and it doesn't learn anything. Okay, so this is just an example. So we have a CLL MRD case. Basically, it's looking at the gates that are already identified by the text in the analysis file and just constructing a template file. And we do this using a Python script, so everything's fully customizable. And basically, it just saves a text file on the server that we can open up and copy and paste and then start to modify in our reports as we see fit. And that's just another example. Okay, so Flowbot is kind of dumb, but he actually does a great job and solves probably most of the cases that we have. Okay, so general steps in applying supervised machine learning. So of course, we need to identify and annotate the training data. So in a lot of cases, this can be the most difficult part. So we need enough data, and we need to actually have a human go in and say, this is this or that. This is CLL, this is mantle cell lymphoma, and so forth. We need to select a classifier, train the classifier, and apply it to a validation or test set. So when we're doing machine learning, we want to separate training data from validation data. So those data sets can never cross, because if some of the test data gets into the training data, it's kind of like cheating on the test for the algorithm. And then we want to ultimately implement it prospectively to new data. So data annotation. We want to develop tools that can present the pathology reports to us that we can then annotate. Another option is we can look at extracted ICD-10 codes from the electronic medical record. So if you're not familiar with ICD-10 codes, basically whenever we file a report, we have to insert ICD-10 codes. And usually these refer to the diagnosis, but not always. Sometimes this is the suspected diagnosis. So those aren't always very trustworthy for machine learning, even though a lot of people use them. And of course, other approaches. So I like to try and be creative about doing the least amount of work to annotate the data as possible. Some tools we have available though are Label Studio. We can use natural language processing. So this is what Label Studio looks like. Basically, you can just use this software that you can preload it with text files corresponding to whatever flow cytometry cases you have. You can highlight various parts of text. And then you can train a classifier to find those texts in additional files. And you can maybe just sign off yes or no that that annotation was correct. So in that way, it can speed things up. This was just an example case where we were trying to look at how we're doing with CD269 reporting. And we found that it identifies the positive and negative reports quite accurately. Okay, so one of the first forays into using supervised machine learning for me was actually applying it to classic Hodgkin lymphoma. So we had a panel that it's not used very often in clinical laboratories, and it kind of takes some training for new hematopathologists to be able to interpret the data. So we were thinking, well, what if we had a machine learning classifier that could look at the data and say yes or no, and maybe that could help new hematopathologists who might be trying to interpret the data. Okay, so what was our approach? So we decided to develop a machine learning algorithm that can predict the diagnosis based on the flow cytometry. We also wanted to design it such that we could apply the interpretability algorithms that I talked about earlier. So we can try and understand how the algorithm is working. So our approach was to create a series of two-dimensional histograms as inputs to the algorithm. And then we would use an ensemble of convolutional neural networks as the classifier. So in terms of data, we took data from about 1,200 cases, 321 positive or suspicious for classic Hodgkin lymphoma, and 921 were negative. And 80% of the data were used for training, 20% for testing. So this is the actual algorithm. So the first step is to take the FCS file, and then we made a projection of histograms. And why did we do this? Well, we wanted to maintain the interpretability. So we kind of thought, well, we'll think about it how a hematopathologist thinks about it. It's not quite the same because we're not subgating histograms and whatnot, so forth here, but it's similar. So then we apply what's called a convolutional neural network algorithm. And this is a machine learning classifier that basically is good for looking at images and making a prediction. So basically, that's all it's doing. And then it spits out a prediction score for each of those two-dimensional histograms. And we can then take the outputs from each of the individual convolutional neural networks, feed it into a final classifier, which in this case was a random forest classifier, and that will spit out the final prediction score, whether it's positive or negative for classic Hodgkin lymphoma. Okay, so it's intuitive, it's flexible. Flexible in that we can also apply additional data to the random forest classifier if we want to, besides just those inputs. And it's amenable to the explainability tools, which I'll explain in a minute. Okay, so the first step is to train the neural networks. So basically, we have to take all the raw data FCS files, convert them to the two-dimensional histograms, and train a convolutional neural network. How can we know whether it's being trained correctly? So we can look at how accurately it predicts the diagnosis for the number of cycles that it's been trained. So each cycle is basically called an epoch or epoch. And basically what that means is it's presenting all the data to it this many times. So if it's epoch 100, it's seen all the data 100 times. And each time, hopefully, it's getting better, tweaking the little internal parameters to be able to recognize positive versus negative. Now, after you train it for too long, it starts to become a little overtrained. So it starts to recognize the training data. It's kind of more like it's memorizing the answers to the test without having the general understanding of how it came to the correct answer. So what we do is we cut off the training after about 50 epochs in order to keep it from being overtrained. Okay, so what happens when we do that? We can look at all the outputs from those convolutional neural networks. And if we plot it as a 2D dendrogram, we can see kind of how the individual convolutional neural networks are predicting the final diagnosis. So on the left here, we have the actual diagnoses. So red is negative, blue is positive. We see over here in the outputs from the convolutional neural networks, we see a lot of yellow. So that's higher probability that it's positive versus darker blue that it's negative. So one thing that was curious was that a lot of the two-dimensional histograms are actually predictive. So we originally were thinking, oh, some of them will be much more useful than others. But in fact, the algorithm is picking up on subtle differences in basically all of the histograms, and it's making a prediction. Okay, so what we can do is then take all those outputs, pass them through a random forest classifier, and make the final prediction. So basically, each of these rows is passed to the random forest classifier. Okay, so for the final results, we had about 89% accuracy, and the area under the receiver operator characteristic curve was 93%. Sorry, 93%, so that was actually quite good. Over on the right, I have plotted what's called a precision recall curve. So this is a common kind of curve that's used in machine learning. Basically, we want the curve to approach the upper right corner. So the farther to the upper right corner we get, the better we say the algorithm was at making predictions. And in this case, it did quite a good job. F1 score is also another metric used commonly in machine learning, and the F1 score here was quite respectable. Okay, so that's great, but how do you explain it? Okay, so we can break the process up into parts. So we can look at the final integrating random forest, and we can also look at the convolutional neural networks. And what we used was SHAP values. So again, what are SHAP values? So this SHAP stands for Shapley Additive Explanations. Basically, it calculates the marginal impact of each of the individual data elements on the final prediction scores. So what it does is it takes the input data and it starts tweaking all the values a little bit, and it sees how does that tweak change the final prediction score. So for the values where a small tweak changes it a lot, that is determined to be a very impactful data point. So we can map back to the important parts of the data for the individual predictions. And it's nice because we can do it not only just for the general picture, but for individual cases. So one case versus another. So when we did that for the random forest classifier, we can look at which of the two-dimensional plots was most useful for predicting Hodgkin lymphoma versus not. And we see in this case that the CD40 and CD15 histogram was the most predictive. And that's quite interesting because CD15 and CD40 can be expressed in Hodgkin lymphoma. What was also interesting was the CD, let's see, can move this. So anyway, down lower, you can see that in one of the histograms, when it was positive, it was really positive. And so that's also interesting for understanding how the algorithm is working. That's the CD30 versus forward scatter. So Hodgkin cells, incidentally, are positive for CD30. Okay, so it can also highlight the key regions in the individual histograms. And we can actually map those back to the single cells. So can we correlate that to individual cells and plot it in normal flow cytometry software? And yes, we can. So basically what we did was we integrated all those SHAP values from all the individual two-dimensional plots and associated those SHAP values with individual cells. And so then again, we just created new channels in our FCS file, which we can then plot using our standard flow cytometry software, and then color the cells according to their SHAP values. And that allows us to highlight different parts of the data. So one thing we notice is that, for example, the lymphocyte population is quite important for the classifier. And so that makes sense given that the lymphocyte background population is altered, or, the cells may have increased CD7 or a greater population of CD4 positive cells in association with classical Hodgkin lymphoma. So that's just another way we can look at and get insight into how the machine learning classifier is working. Okay, so I basically mentioned this. So another interesting point also is that this is an unbiased approach to looking at the data. So basically, it might find populations that we were overlooking as pathologists that might actually be predictive of whether classic Hodgkin lymphoma is present or not. So that's pretty exciting for a basic science point of view. Okay, so which machine learning algorithm is best? Okay, so actually, we decided to compare 3 machine learning algorithms in this study, but there are more out there, just to mention that. And we decided to compare accuracy, speed, and interpretability. And the 3 we chose to look at were Ensemble CNN, FlowCat, and FlowUmap. So basically, I'll describe, so you already know the Ensemble CNN approach is what I just described. FlowCat is basically an algorithm developed by Wolfgang Kern's group at the MLL in Germany. And then the last is basically plotting a UMAP, taking the UMAP as input to a machine learning classifier and making a prediction. So the FlowUmap did not compare very well, so I won't mention that. But the other 2 I'll mention. So the FlowCat, so how does it work? So basically, FlowCat calculates flow sums for all the input data. And then what it does is it takes all those nodes as the inputs for the final machine learning classifier. And so in this case, they used a convolutional neural network at the end, but other groups have done similar things and used like a random forest and so forth. So what did we do? We took some open data, which was previously published in association with FlowCat. And basically, it was a set of B-cell screening FCS files that were looking, and they were labeled or annotated for normal CLL, marginal zone lymphoma, and so forth. And it consisted of 3 panels that were used as inputs. We followed a similar training or testing split as was previously used for FlowCat. And we split the data for the training, the Ensemble CNN. Part was used for the CNNs and part was used for the random forest. And what did we find when we compared the 2? So basically the accuracy was almost indistinguishable for the two classifiers. And so what do we take away from that? So actually, this speaks to the importance of the quality of your data. So if you have a representative data set, it might not matter so much which machine learning algorithm you're actually using, because at a certain point, the accuracy is not improving a whole lot. So if you're not really sure which machine learning algorithm to use, I would say just pick one that you can get to work and start with that and go from there. Okay, so there was a difference in the time required. So the FlowCat did take a fair amount longer time to process the data. And we can also use the same SHAP approach for interpreting the data, but in this case, we're looking at multiple classes or labels versus just two. Okay, so just, let's see, it looks like I'm out of time. So I'll just go quickly through this last example. So we also were interested in, can we use the same approach to predict whether we need additional antibody panels? And can we use that to help improve or speed up the workflow in our lab? So the use case we decided to look at was chronic lymphocytic leukemia. So that was the most commonly used additional antibody panel in the lab. And we were basically using it to help distinguish between chronic lymphocytic leukemia and mantle cell lymphoma, because these are both CD5 positive neoplasms. And basically, for a given set, or for a new case, techs would automatically choose the panel as it was needed. If the techs knew that the patient already had a diagnosis of CLL or mantle cell lymphoma, they did not add the panel. So what did we do? We basically took retrospective data and trained the classifier using non-discernible CNN. And we basically came up with a very accurate classifier. Okay, so that's great. When are you going to actually apply it in real time? So we already did that as well. So basically, we had a workstation checking the server. So every 5 minutes or so for new FCS files as they were deposited. And then it would look at those FCS files and make a prediction. And if the probability of needing a panel surpassed a certain threshold, it sent an email. And basically that email came to me. Okay, so prospectively, this is what it came up with. The receiver operator characteristic curve was actually quite good. The area was 0.89. Precision recall didn't look as good, but still decent. Okay, so the cases it looked at, it got 17 that were called false positives and 5 false negatives. So of the 5 false negatives, 3 cases did not have the CLL panel included in the final report. So for whatever reason, the hematopathologist decided, I'm not going to even try to bill for this because it probably wasn't important or useful. One case was suspicious for a small number of CD5 negative large B-cell lymphoma cells. So it wasn't clear, maybe that was ordered in error. One case was CSF involved by mantle cell lymphoma. So it would have been justified, but it wasn't run, perhaps because there weren't enough cells to run another panel. And of the 17 false positive, 13 had prior diagnoses. And so if the algorithm had known that, it wouldn't have predicted another being needed. So in fact, when you look at all the details, it performed extremely well. Okay, so some of the challenges in applying machine learning to flow cytometry. So right now, a lot of regulatory uncertainty. The FDA has basically made it difficult to now have laboratory developed tests. So every test performed in a hospital basically will soon need FDA approval. And so this is a very big regulatory hurdle. Data availability, and then also generalizability of the algorithms. So if we train something in one lab, can we apply it to another lab? And in most cases, the answer is no. Okay, so just in summary, there are many potential applications for machine learning and AI and flow cytometry. It can assist in identifying diagnoses, predicting add-on tubes, and so forth. And the interpretability tools can help us in understanding how the algorithms are performing. And of course, don't overlook the easy ways. So for example, Flowbot, which actually does a good job for many applications. And I'd just like to acknowledge some of the people at Weill Cornell Medicine, including Luigi and so forth, some people at the University of Washington, and David Ng at University of Utah. So thank you very much.
Thank you. Do we have any questions?
Hi, great presentation and a comprehensive overview of most of these things. My question is, on a day-to-day basis, the fluorescence shifts, right? You know this. How does CNN get used to this or normalize this or deal with this when they makes its prediction?
Yeah. So one approach is just to have a lot of training data that might contain those shifts. The other factor why I think it performs well is that it's using operations inside the convolutional neural network, where it's doing what are called max pool operations. So it's looking at all the pixels in an area, and then it's not just looking at individual pixels, but it's looking at the areas to see how they compare with each other. And so I think because it's doing those kind of max pool operations, that's helping mitigate those problems.
Just a quick one about how you get your training data for this. Do you find that if you don't have balanced training groups, meaning 50% Hodgkin's positive, 50% Hodgkin's negative, do you think that will have an impact on the overall accuracy of the model that you're generating, and it's likely to over or under predict one condition over the other?
Yeah, so what we do is we actually balance the data by basically repeating the underrepresented examples. So you can do that either by just selecting parameters within most machine learning classifiers to say you should now balance the data or the number of times each one's getting presented to the classifier. Actually, interestingly, for whatever reason, we found that we actually needed to just actually duplicate the data. And because for some reason it seemed like it wasn't working with the automated balancing. So just a no.
Thank you so much. Thanks.